Hello everyone, myself Anishura TS, first MSc Biology. Today I want to discuss about the factors that influence microbial growth, environmental and nutritional factors. Microbial growth. Microbial growth refers to an increase in cell number and cellular constituent rather than increase in cell size. It depends upon the ability of the cell to form new protoplasm from the nutrients available in the environment. The increase in the bacterial cell numbers occurs when the microorganisms reproduce by binary fission. Binary fission begins when the DNA of the cell is replicated. The growth curve. Here, the graph shows the four phases of the microbial growth: lag phase, log phase, stationary phase, and death phase. The first one is the lag phase. During lag phase, there is no increase in cell number and mass, but the cells is synthesizing new components. In the lag phase, the microbial cells are not dormant. The lag phase varies considering in length with the conditions of the microorganisms and nature of the medium. Second one is the log phase. It is also known as exponential phase. In this phase, the number of cells increases exponentially with the time. During the log phase, microorganisms are growing and dividing at maximal rate possible given their genetic potential, using the nature of the medium and the conditions under which they are growing. Their rate of growth is constant during the log phase, that is, the microorganisms are dividing and doubling in number at regular intervals. The rate of exponential growth of the microbial culture is expressed as generation time. During the exponential phase, each microorganism is dividing at constant intervals, thus, the population will double in number during a specific length of time called generation time or doubling time. Let the n0 is an initial population number and nt is the population at a time t, and n is the number of generation in time t. That is nt, the population at the time t, is equal to n0 into 2 raised to n. The number of generation per unit time, that is k, is equal to n by t. If the population is doubled, then nt is equal to generation time g. By applying this in the above equation, k is equal to 1 by g. So the generation time g is equal to 1 by k, that is t by n. Third one is the stationary phase. In a closed system, eventually population growth ceases and the growth curve begins horizontal. In stationary phase, the total number of viable microorganisms remains constant. This may result from balance between the cell division and cell death, or the population may simply ceases to divide, but they remain metabolically active. Microbial population enters stationary phase because of the limitation in nutrients, limitations in oxygen availability, and accumulation of toxic waste products. And the last one is the death phase. If incubation continues after the population reaches stationary phase, a death phase follows. In this phase, the viable cells decline and loss happens in the total number of cells. The factors that influence microbial growth are categorized into two: environmental factors and nutritional factor. First one is the environmental factors. The growth of microorganisms also is greatly affected by the chemical and physical nature of their surrounding. The major environmental factors affect the microbial growth are solute and water activity, pH, temperature, oxygen level, and pressure. The first one is the solute and water activity. The selective permeable plasma membrane separate the microorganism from their environment. They can affect by changes in the osmotic concentration in their surrounding. The microorganisms that place it in hypertonic solution will burst, and that place it in hypertonic solution will shrink. So the microbes be able to respond to changes in osmotic concentration. Microbes in hypotonic environment reduce the osmotic concentration of their cytoplasm, and the microbes that placed in hypertonic environment increase the osmotic concentration. According to the solute and water activity, The microbes are categorized into two groups, that is haplophiles and osmotolerant. The first one is haplophiles. The microbes adapted to extreme hypertonic environment is haplophiles, and they grow optically in the presence of NaCl or other salt at a concentration about zero point 0.2 molar. Example is halobacterium. And the second one is osmotolerant. They will grow over a wide range of water activity or osmotic concentration. Example is Staphylococcus aureus. The second one is pH. pH is a measure of hydrogen ion activity of a solution and is defined as negative log of a hydrogen ion concentration. Each species has a definite pH growth range and pH growth optimum. According to pH, the microbes are categorized into three, that is acidophiles, neutrophiles, and alkalophiles. The first one is acidophiles. Their growth optimum between pH 0 to 5.5. Examples is sulfolobus acidocaldarius. It's survive at a pH of 1 to 3 with high temperature. And a second example is Ferroplasma acidaromanus and Dicrophilus oshmi that belongs to pH 0. The second one is neutrophiles. Their growth optimum between the pH 5.5 and 8.0. And example is Escherichia euglena paramecium. The third one is alkalophiles. Their growth optimum between the pH 8.0 and 11.5. Example is Bacillus alkalophilus and Nitrobacterium. The third one is temperature. At optimum temperature, all the enzymes in microbes are considered together as the rate of each reaction increases and metabolism as a whole become more active and the microbes grows faster. Beyond certain point, further increase actually slow growth and sufficiently high temperature is lethal. The microorganisms can be placed in one of the five classes based on their temperature range for growth. First one is psychrophiles. They grow at 0 degrees Celsius and have an optimal growth temperature of 15 degrees Celsius or lower. Example is Bacillus trichophilus and Chlamydomonas nivalis. Second one is psychotroph. They can grow at 0 to 7 degrees Celsius and has an optimum between 20 to 13 degrees Celsius and a maximum around 35 degrees Celsius. Example is Listeria monocytogenes. Third one is mesophils. Have an growth optimum between 20 to 45 degrees Celsius. Example is Escherichia coli, Trichomonas vaginilis. The fourth one is thermophiles. They can grow at 55 degrees Celsius or higher. Their optimum often between 55 to 65 degree. Example is Diobacillus stearothermophilus and Cynidium caldarium, etc. The last one is hypothermophiles. Hyperthermophils uh, are thermophiles that can grow at 19 degree or above. Their maximum about 100 degrees Celsius. Some of prokaryotes growth optimum between 18 degrees Celsius and about 113 degrees Celsius. They do not grow below 55 degrees Celsius. Example is Sulfolocus, Pyrococcus, etc. The fourth one is oxygen concentration. The importance of oxygen to the growth of an organism correlate with metabolism. An organism able to grow in the presence of atmospheric oxygen is an aerobes, whereas the one that can grow in its absence is called anaerobes. According to the oxygen concentration, they are further classified into five. The first one is obligate aerobes. They are multicellular organisms that completely depend on atmospheric oxygen for length. Example is Micrococcus luteus, Pseudomonas mycobacterium, etc. Second one is facultative anaerobes. These are organisms do not require oxygen for growth but grows better in their presence. Example is Escherichia, Enterococcus, Saccharomyces cerevisiae, etc. Third one is aerotolerant anaerobes. It ignore oxygen and grow equally well whether it is present or not. Example is Staphylococcus pyrogens and Enterococcus faecalis. The fourth one is strict or obligate anaerobes. They do not tolerate oxygen at all and die in its presence. Example is Clostridium, Bacterioides, and methanol bacterium, etc. Aerotolerant and strict anaerobes cannot generate energy through aerobic respiration and must employ fermentation or anaerobic respiration. The last one is microaerophiles. They are damaged by the normal atmospheric level of oxygen and requires oxygen level below the range of 2 to 10 degree 10 percentage for growth. Example is Campylobacter, Spirillum voltanum, etc. The last one is pressure. Organisms that spend their lives on land or on the surface of the water are always subjected to a pressure of 1 atm and are never affected significantly by the pressure. Some prokaryotes live in the deep sea, whereas their hydrostatic pressure can reach 600 to 1100 atm and the temperature about 20 2 to 3 degrees Celsius. Many of them are bacterotolerant. Some prokaryotes in the gut of deep sea invertebrates such as amphipods and holothurium are truly barophiles that they grow rapidly at high temperature. Photobacterium profundum, Schwannella bentica are examples. These barophiles grows more rapid at high hydrostatic pressure. The second one is the nutritional factors. Nutrients are substances used in biosynthesis and energy production and therefore are required for the microbial growth. All the bacteria require several macro and micronutrients. Macronutrients are needed in relatively large quantities while micronutrients are used in very small amount. In addition to the needs of carbon, hydrogen and oxygen, all organic requires source of energy in electron for the microbial growth. On the basis of source of carbon, electron and energy, microbes can be categorized into following types. Based on the energy source, they are categorized into phototrophs and chemotrophs. Phototrophs uses light as source of energy, while chemotrophs uses energy from the oxidation of reduced chemical compounds. Second one is electron source. They are categorized into organotrophs and lithotrophs. Organotroph uses reduced organic compound as a source for reduction potential and lithotroph uses reduced inorganic compound as a source of reduction potential. Third one is carbon source. They are categorized into autotrophs and heterotrophs. Autotrophs can use CO2 as a sole carbon source, while heterotroph requires an organic carbon source. They cannot directly use the CO2 as a carbon source. The fourth one is nitrogen source. There are four types of nitrogens, that is organic nitrogen that primarily from the metabolism of amino acid. Second one is oxidizer forms of inorganic nitrogen, that is nitrites and nitrite. And third one is reduced inorganic nitrogen, that is ammonium. And the last one is the nitrogen, that is dissolved nitrogen gas, that is by nitrogen fixation. The fourth one is the nitrogen source. There are organic nitrogen, that is preliminarily from the catabolism of amino acid. Then oxidizer forms of inorganic nitrogen, that is nitrate and nitrite. Reduced inorganic nitrogen include ammonium, and the last one is dissolved nitrogen gas, that is by nitrogen fixation. Fifth one is phosphate source. There are organic phosphate and inorganic phosphate. And then sixth one is the sulfur source. The organic sulfur, oxidized inorganic sulfur, that is sulfate, reduced inorganic sulfur, that is sulfide, and elemental sulfur. Then other nutrient requires are amino acid, nucleotide bases, and enzymatic cofactors or vitamins. That's all about the microbial growth. Thank you.